So, overview of the study. This study investigates how exposure to cat odor affects the behavioral and physiological responses of cats. Specifically, it examines four things: immediate responses to cat odor, which is unconditioned because they have not yet been linked to any object. So, immediate responses. Day 2, conditioned responses to a previously cat odor associated environment or stimulus. Day 3, changes in locomotor activity, heart rate, and blood pressure as indicators of anxiety and fear. And how rats engage in defensive behavior such as head out behavior, flat back approach, and vigilant rearing in response to predator odor. Right, so key results and interpretation. Immediate behavioral response to cat odor. Day 2, unconditioned fear response. When exposed to cat odor for the first time, rats displayed robust defensive behaviors, including head out behavior, which is staying in the hide box but keeping the head out or head or shoulders out for vigilance and for scanning the environment for any potential threats, like a cat in the nearby environment. Right, so that's one, head out behavior. Then second is decreased locomotion and exploratory activity, since it knows that since there is cat odor, there might be a potential threat, there might be a cat nearby, right? So that's the second one. Then we have the flat back approach, approaching cautiously in a low posture to minimize detection by the threat. And fourth is increased blood pressure but no significant heart rate change. So these are the defense mechanisms that it uses, defensive behaviors, right? And interpretation, learning, and memory. These behaviors reflect an innate and unconditioned fear response, right? Meaning they do not require prior learning to this. This is innate, right? Inborn in them. So this response is an evolutionary adaptation that instinctively recognizes predator orders as a threat. Now, the high BP response without a heart rate increase suggests that stress-induced autonomic response are selective and context-dependent. The fact that rats did not freeze but instead engaged in active avoidance strategies suggests that access to a hide box allows for risk assessment behavior, influencing the expression of fear responses. Second, conditioned response to context previously attached or associated with cat odor. Day 3, right? So when placed in the safe test chamber with a previously unworn cat collar, that is no fresh odor, rats still exhibited reduced locomotion and exploration, indicating a learned fear response. So this has become a conditioned stimulus, right? Now, blood pressure remained moderately elevated, showing a physiological sign of conditioned anxiety. Heart rate decreased significantly, possibly due to the reduced emotional stress compared to day 2. Behavioral changes on day 3, similar to day 2 but milder, confirmed that the presence of contextual fear conditioning, the rats remembered the association between the chamber and the previous cat odor exposure. Now, interpretation and learning and memory. This demonstrates associative learning, where the rats formed a context-dependent memory linking the test chamber to the previous predator odor. The conditioned fear response is an example of Pavlovian classical conditioning, right? So the unconditioned stimulus US is the cat odor. Unconditioned response is fear behaviors like hiding, vigilant rearing, head out approach, and reduced locomotion, increased blood pressure. Then the conditioned stimulus is the test chamber or the collar, CS, and the conditioned response, CR, is again fear behaviors despite the absence of the cat odor, right? So the fact that heart rate decreased while blood pressure remained elevated suggests that different physiological responses are involved in fear memory process. Neural mechanisms. This learning likely involves the amygdala, fear processing, hippocampus, contextual memory, and prefrontal cortex for risk assessment. Right? Now, differences between cat odor and other predator odors, fox TMT. So, unlike fox-derived odor TMT, which had no effect on locomotion in prior studies, cat odor significantly reduced activity. This suggests that not all predator odors are equally potent. Cat odor appears to be a stronger fear-inducing stimulus than fox odor to rats. Right? Now, it may be different for other animals. For example, for fox, in the case of fox hunting a raccoon, fox is the predator and a raccoon is the prey. So they would be less scared by a cat and more scared by a fox. So for them, for a raccoon, the fox's odor would be a stronger fear-inducing stimulus than the cat's odor, right? Now, interpretation in learning and memory. This highlights stimulus generalization versus specificity in the fear learning. Rats likely distinguish between different predator odors based on their evolutionary history. They may encounter foxes less frequently than cats, right? Distinguish between different predator odors based on their evolutionary history, right? Stimulus generalization versus specificity. The degree of conditioned response, stronger for cat odors, suggests that learning is shaped by natural predator-prey interactions. Now, role of defensive behaviors in learning and memory. So, head out behavior, vigilance. Rats spend most of the time in the hide box with their head out rather than fully concealed. This suggests a risk assessment strategy rather than full avoidance by scanning their environment for potential threats. Memory mechanism. This behavior aligns with Blanker's theory of partial predator stimuli, where animals use cautious approaches when dealing with indirect threats, right? Partial predator stimuli. Cautious approaches when dealing with indirect threats. It supports the idea that fear learning is not just about avoidance, but also about adaptive decision making. Right? The hippocampus is likely involved in spatial awareness and contextual memory retrieval. Then we have the flat back approach, stealth. This reduces visibility to predators while still allowing approach. It is a form of learned adaptive behavior seen in other fear-based paradigms. Right? Memory mechanism. This behavior can be linked to the basal ganglia and the prefrontal cortex, which are involved in habit formation and motor control during learned fear responses. Now we have physiological responses and learning. So blood pressure as a more reliable fear indicator than heart rate. So blood pressure remained high despite changes in activity. Heart rate only dropped on day 3, suggesting a delayed autonomic response. Memory mechanism. Amygdala-driven fear responses might elevate blood pressure independent of movement. Heart rate variability may be modulated by the parasympathetic nervous system, which interacts with emotional learning. Now conclusion, how this study relates to learning and memory. So unconditioned learning Initial exposure to cat odor triggered instinctive fear responses, head-out behavior, hiding, and reduced locomotion. Classical conditioning. The test chamber became a conditioned stimulus, eliciting fear even when no odor was present. Test chamber became a conditioned stimulus, eliciting fear even when no odor was present. Basically, right? So the hippocampus and amygdala are key in forming and retrieving these fear memories. Adaptive decision making. Instead of freezing, rats engaged in risk assessment strategies, showing that fear memory involves context-dependent decision making rather than reflexive avoidance. Physiological learning markers. Blood pressure remained elevated, making it a better indicator of conditioned anxiety than heart rate. Heart rate changes were more complex, reflecting the interaction of movement, stress, and habituation. Predator odor learning is specific. Rats responded more strongly to cat odor than fox odor (TMT), suggesting species-specific learning based on evolutionary experience. So final takeaway, instinctive responses, unconditioned fear, associative learning, contextual conditioning, decision-making strategies, risk assessment versus avoidance, physiological changes, blood pressure and heart rate regulation. So that's it about this video. Thank you.